Thank you for having us and welcome everyone. Just as was mentioned, we want to spend a few minutes really kind of discussing some work we've done as part of the Intrepid Alliance in developing an antiviral target compound profile for pandemic preparedness, primarily viruses that have pandemic potential. So myself and Jim will be presenting today. The topics we're going to talk about, and just very briefly, I'll just mention what exactly the Intrepid Alliance is. I'll talk a bit about some of the activities we've been doing, which have occupied a fair amount of our time over the last 8 months. And that was performing a landscape of the antiviral clinical development, what's going on out there. And again, we just use publicly available information going forward. So we don't use any kind of proprietary databases for this. But as part of that work, we've been looking at target TTPs and target product profiles, as well as by looking at that, we're interested in getting more details related to the compound profile in the preclinical space as we develop new drugs. So the next slide just lists out a couple of facts about the Intrepid Alliance. It came up as part of reviewing what occurred during COVID. It is a consortium of 7 pharmaceutical companies that are listed there. Again, this is a nonprofit organization and is a significant scientific contributor to the 100-day mission and the International Pandemic Preparedness Secretariat, which came out of the G7. And really, this is a call to action of really having a specific number of antivirals ready to go into development if a new pandemic is occurring. We'll get into more specifics about that in a second with that. So as part of this activity, I think it was important to kind of understand where we are and to form a landscape of what drugs are there. Obviously the mission is to try to find, if you could go back to the one slide there, I'm sorry, is to just identify phase 2 ready drugs ready to be deployed into clinical trials following the identification of a pandemic. We do concentrate primarily on antivirals, and these are direct acting antivirals. We really don't include in our landscape those compounds that affect host targets. The next slide just lists some activities that we've done over the last year. We had a large antiviral summit about a little more than a year ago. Number of stakeholders came and really coming together, and it's really the emphasis on collaboration. How can we work together to bring forward antivirals quickly in the face of a new pandemic? So we've been primarily, as I said, working on our landscape. If we should show the first next slide, Jim. This was our initial look at clinical compounds. So these are products or compounds that have made it into human testing from phase 1 all the way to approved products. And you see here from our survey, the mix, we concentrated on 12 viral families. Again, there's always emphasis on what are the priority pathogens. We're waiting to hear from the World Health Organization what exactly those are. But I think of these 12 families will include most that are important for pandemic likelihood. So they're listed there along the bottom. You look at the number of compounds or percentage of compounds. And what you're struck with quite a bit is the emphasis in coronaviridae as well as orthomyxoviruses. So really the COVID flu compounds dominate the scene. And looking at across the other 10 viral families, the numbers of compounds at various stages, they tend to be somewhat at the earlier stage, are much less going forward. So if we can look at the other slide, this is actually on our website, and we welcome you to look at it. It's intrepidalliance.org. We have a large slide set with it, which are free to download and use as slides. We put together this wall. It's actually interactive, and I can't show it here. But if you click on each of the specific compounds, you can see the developer, the mechanism of action, what virus that it's going for. And so I think it's useful to kind of see where we are right now. And this is as of March 2024. We will be updating this on a quarterly basis. And so the next one will come out in end of July. We're also working on preclinical compounds. And so that will also be shown for the first time coming forward in July. I will say as a preview of that, the emphasis again on coronaviridae as well as flu viruses is there. And so really there's a real emphasis in those areas. And so in other viral families, there's much more of a gap going forward. And again, to emphasize, these are specific antivirals, and it is public information that we're using to construct these landscapes. So going forward, just the final slide I just want to show as part of the introduction. As we look forward, how do we speed along the development? Clearly, target product profiles are quite helpful. They serve as a overlying roadmap of how we want to get to and what kind of aspects of the drugs contain that a patient can actually take it. And so we really make it very patient-centric going forward, and it covers a whole host of areas. And one of the things we did initially as part of the Intrepid Alliance, we took a look at the existing target profiles that are out there, one of which is from the NIH and AViDD for pandemic preparedness type viruses. And we took a look at it, and I think one of the things we found, and all these target profiles are excellent, but we did find that there was not a lot of information on earlier stages or more of the preclinical part of the development process. So often you'd say for the TPP, you'd say safety, and they would just say the drug's safe. Well, what does that mean? So we felt that there was a need, or we wanted to put together more specific clinical characteristics. And so hence that led to the development of our target compound profile. And Jim, I'll turn it over to you, and we'll go through kind of the specifics with it. But again, we're presenting this not as something in hard cement, everyone needs to follow it. We really put it in the spirit of collaboration. We'd like to hear what people think about it, what's helpful, what's not. It's also meant to be flexible. And so again, if you're talking about where time is of the essence, perhaps some aspects you can, not worry about as much as others. But I thought that what we thought was to really have these characteristics laid out would be important for people to use as part of their thinking as they're bringing forward new antivirals from early discovery. So with that as an introduction, I'll turn it over to you, Jim.
Okay, thank you, John. And as as John mentioned early on, this really was an effort of subject matter experts, if you will, from all of the companies of Intrepid Alliance working with us. And also we engaged with a few different external drug discovery developers, both academic and biotech types of entities, to really get a barometer on the level of information and what have you. So recognizing that each company, each academic group, each entity may have its own specific things, we tried to come to a bit of a consensus, but also try to walk that fine line between how much information or criteria do you provide that would be helpful versus too specific. And so we've tried to strike that middle ground. But basically it's an industry standard preclinical view, if you will, based upon the years of experience of developing antivirals from the different companies. and looking at, what are the main things that you at least you want to have on your radar as you're going through a discovery and early development program. And it's, again, as John mentioned, it's really a guide to also be amended frequently. Again, this isn't like stone tablets coming down from Mount Sinai, and this is what it is. This is really, this is how we go forward. And for certain viral indications, there may be certain criteria or characteristics that are more important than others, or perhaps, for example, the inclusion of pediatric formulation work early on may be more important for others. And again, we really value taking a patient-centric view, considering the ultimate intended use. And with that, you're looking at, prevention, treatment, you're looking at, is this a single agent? Is this combined with other agents? Polypharmacy, how is the dose administered? And who are the different target patient populations? Because rarely is it one patient population. And how do you factor in those criteria into the drug characteristics? And then we looked at it in a way that these criteria can be used to identify potential clinical candidates with a focus on the pandemic causing agents. And then also with the pathway to an IND, because it's not enough to dose 12 mice in a initial research study and none of the mice die, and you say, okay, well, yeah, that's safe. Well, that's a good sign, but really to get into the clinic, as I think everyone knows, you need a really robust preclinical plan to evaluate. So we've tried to capture different criteria based upon regulatory guidance from FDA, from EMA, and others, as well as industry input. And so basically what we boiled down to were 6 key categories. And within these categories, there's some specific criteria that we've outlined. We do have a manuscript version of this that we're submitting for publication that has much more of the granular details. But we'll walk you through 6 slides here, one for each of these, just to give you a feel for the types of information and the level of detail that we'd be looking at. So in the first instance is potency and efficacy in vitro. And what the very first thing we all agreed on was mechanism of action is really important, because you want to have a known target and a known mechanism of action that's supported by evidence from biochemical or cell-based assays. Rather than something that, well, we're not quite sure how it works and it may work this way, and then it leads to lots of other potential questions and potential safety issues as well. And then, of course, you're focusing initially on your virus of interest, but you're also looking at spectrum of activity within a virus family and potentially across virus families. We also recognize the dynamic challenge that we have in terms of the more you try to develop something to be very, very broad spectrum, sometimes you lose potency against certain viruses as you do that. So you're always trying to balance one thing against the other. But at a minimum, you're looking at the virus of interest, activity against clinically relevant isolates that are circulating or could be circulating. You're doing experiments to look at combination use. Now, the in vitro 2-drug combination assays are merely that. They can give you an idea of, are these antagonistic or are they not antagonistic? You can debate synergy and additivity, but in the end, the clinic tells you the reality of combination benefit when things are used together. But the in vitro work that can be done can inform how you would use this as a potential combination therapy. And this is a different characteristic than drug-drug interaction profile, which we'll come to later. And then for resistance, of course, it's a major concern for any antiviral and antimicrobial agent. And so, we did try to come up with some parameters But as you would imagine, each virus has its own thing and certain mechanisms are there. So, I think just that it's a longer time to get a resistant variant in vitro than what the clinical time would be considered. And then also having activity against circulating variants with polymorphism so that you reduce the risk of inherent resistance in the clinic. And then cross resistance, depending on the situation, you may have access to viruses that have been emerging as a result of, for example, the monoclonal antibody drug pressure. Does it work against those, for example? And then in terms of in vivo potency and efficacy, now one can debate whether you have to have this information, but it's very valuable, especially in these situations with unknown viruses or emerging viruses. But also you want to be designing your experiment and your criteria and your attributes in a way that addresses a prophylaxis use for infection or a treatment of infection, and then also prophylaxis for blocking transmission. And we did suggest some different um, markers, if you will, for the amount of reduction in incidence of infection or viral load that would be considered. And we have more details in the full target compound profile. But here again, it's just trying to give a guidance of what level of activity do you want to see that would give you more confidence that this compound has drug-like properties and should move forward to the clinic. We also acknowledge that animal models are models and they may not directly correlate with responses observed in clinical trials. However, again, they do have utility when used to help define efficacy as well as PK-PD relationships, et cetera. In terms of preclinical pharmacology, we did try to capture in different attributes obviously the PK profile of the drug itself, also the PK/PD relationship, different measures of drug metabolism, drug-drug interaction profile potential, and then tissue exposure, which may be paired with the in vivo efficacy models as well. And with that, what you're looking at with small molecules, you need to have two species, rodent and non-rodent usually. And we suggest that, focusing on something that's twice daily oral dosing is, probably more favorable for a patient, ideally once a day, but, you may not be able to achieve that on a first generation. We also wanted to factor in the PK/PD relationship, the activity related to the exposure and the antiviral activity. And some folks have called this the PK/PD index value. And another thing that comes into play here is it can get a little bit tricky if you only rely on in vitro antiviral efficacy, because you may have one compound that's more efficacious in vitro, a lower IC50, et cetera. But its drug-like properties or its PK properties require that the dose is 3 times a day or something like that. You could have another compound that's less potent in vitro, but its properties are more favorable. And actually that compound would be better to bring forward because it's about, again, the ability to dose a patient and get sufficient exposure for activity. And then the other characteristics below are really looking at the things we know that we're looking at not only measures of clearance and metabolism, but you're looking at CYP induction, CYP inhibition. This gets into the drug-drug interaction profile, which again is distinct from combination therapy treatment. And then looking at disease-relevant target tissue exposure, is it similar or better than blood plasma? These are things that might inform, especially if you're looking at more than one compound or a different series, this might inform which compounds you might or series you might take further along. as John alluded to, in terms of preclinical safety, there's numerous regulatory guidance materials and requirements from the FDA and EMA and others that really define what they want to see. But what we tried to do was take that information as well as the collective experience and say, what are the main things that at least you want to check the boxes for if you're able to, as early on as possible. And so dose range finding experiments in rodents and higher species, these can be used to support dosing. And then you get into your GLP IND enabling program using more appropriate exposures in that way. And of course, those programs are more costly and more involved. Repro and JuviTox studies planned, some rather than kick it down the road, do it earlier than you can, but it may be that it's planned early on, but at least it's on the radar as opposed to something that's way later. There is regulatory guidance about the metabolites of greater than 10% that you have to look at, and so that we included that. In vitro genotoxicity, we understand some the complexities in terms of having different assays and different measures, but in general, we at least wanted to provide some suggestion regarding being negative in an Ames assay is certainly a target goal, and also in the chromosome aberration and micronuclease assays. Cytotoxicity in a variety of cell types, you want to be able to have that at a good level so that your antiviral activity in vitro preclinical is not because you're killing cells or affecting that way. It's really safer in that regard. Off-target activity, again, there's these panels and ion channels, et cetera, that one can run and have access to. And looking at, I think at a minimum, ideally, you want to have none with a 10 micromolar off-target activity, and upside is you'd be down in the 1 micromolar range. These numbers are debatable. Again, this is just an evergreen guidance to say, relative amount of that where you're looking at to minimize the off-target effects. And then, of course, HERG and the BSEP assays, looking for effects there. These are values that we suggest based upon the different experience. But again, these are open to input from regulators as well for a given scenario. And then it's CMC and chemistry. This is really important because it also speaks to speed of achieving what John referred to, the 100 days mission. If you have something that could be available, but it takes many months to produce it, then you're out of the ball game, so to speak, in that regard. And also, we're not really commenting on the cost of goods at this point in time, but the point being the criteria, the characteristics of the drug, the API, et cetera, would be such that you could have a good solubility profile, the minimum complexity with raw materials, et cetera, a minimum number of steps, ideally less than 10 steps, because again, this gets to your ability to have speed of manufacturing if you need to ramp up. But also importantly, because again, we're thinking pandemic preparedness, we're thinking a variety of geographies, right, as well as patient groups. And so stability, having ambient temperature supporting zones 1 through 4. And then, as we mentioned, pediatric formulation work for certain viral infections. You may want to be doing that work earlier, much earlier, maybe even in parallel with the adult formulation because of the nature of the affected population. But at a minimum, you want to have that planned and starting ongoing earlier rather than something you would add on later after you get approval for an adult formulation. And so I think these are important because it speaks to, again, speed to manufacture in the case that we need to ramp up something in the face of a pandemic. And it speaks to the global patient population having medicines that are accessible to them. And then finally, we're not really about diagnostics. There's the group FIND and others that deal with that. But we have to be aware of the role of diagnostics in the use of our medicines that we're developing. And, these are three just simple areas. One, there's probably others, but that we just wanted to highlight that, you want to be supporting in any way you can the development of those folks who are working on assays that diagnose the infection, because obviously that helps better pair, right, the appropriate use of the medicine. Um, Ideally, it's a short turnaround time readout, and also it's validated to certain regulatory standards so that it's not a lot of false positives. Informing the use of new medicine, we wanted to capture this because, as we know, with not only polypharmacy, but also, patients with, for example, renal or hepatic disease, you want to understand more about, not only the characteristics of the drug, but in those patient populations, are they at places where we will know that those individuals are there, or if there's a change in their disease paired with the medicine. And then finally, the virus susceptibility assays. These are really ones that evolve as you get more information by genotype and potentially phenotype, and you start getting more information on natural evolution, polymorphisms for immune pressure, vaccine pressure, antibody pressure, and then you get into drug pressure leading to changes at specific drug targets. And so again, we think to just be mindful of how you might partner with, or what might be required for the use of a compound with a specific target. Would you need to have a special assay available, or is the standard assay appropriate in that regard? Uh, so that really just sums it up. Again, we're looking at this in a way that it augments the existing materials that are there, just goes a little deeper into some of the nuts and bolts of preclinical types of criteria, and serving as a guide. And again, evergreen, it could be amended, it could be modified. It's a very collaborative project, if you will, but ultimately it has to have a global patient-centric view, considering the ultimate intended use in order to have, best value added of it. So I'll pause there, turn it back to John, and then I guess we could, I don't know if you want to do a couple questions.
Okay, well, thanks, Jim. And as was said, that comes to the conclusion of our presentation. I know we're a little bit long with it. We'll be glad to take any questions. I do see one Q&A was asking about the 5 Rs of drug development that AstraZeneca has written about. This is like the right target, right tissue, right patient. And so essentially, we're part and parcel with it. You can find all the R's in our target compound profile, but this is really meant to be the more details, the real more specifics of the cookbook as you're doing that forward. So what we look at it as totally fitting with the kind of the 5 pillars or 5 Rs or whatever you want to call it as you go forward. And we also look at this as a companion piece, say, with the target product profiles that the NIH has published on their website for pandemic viruses. We do want to make it more specific for those viral pathogens. But again, that's the overall principle here.